Good morning, everyone. Our lesson today will be about measuring biodiversity. The learning objectives include: number 1, understand how biodiversity can be compared in different habitats using the formula to calculate an index of diversity. And we are going to learn how to use this formula to calculate the index of biodiversity. 2, know how biodiversity can be measured within a habitat using species richness within a species using genetic diversity by calculating heterozygosity index. Also, you're going to learn how to use this formula to calculate the heterozygosity index. 3, understand that changes in allele frequency can come about as a result of mutation and natural selection. You're going to learn some new terminology: diversity index, genetic diversity, heterozygosity index, and allele frequency. And then you are going to answer checkpoint questions in your textbook, as long as with some past paper questions. Let's get started.
Oh. A wild classroom of eco-geeks exploring biodiversity. The world is a beautiful mystery, life learning and discovery. Eco-geeks.
This world has many, many species. Have you ever awoken from a night of, say, sleeping in a hammock in the jungle to ponder what biodiversity is? Okay, my guess is probably haven't, but we do all the time. Questions like how many species are there in the world, or why really is it important to protect all these species, come to mind.
So in this video, we thought we'd touch on a few of the major topics of biodiversity.
Oh wow, what a place to wake up. What a great place to search for biodiversity. In fact, if you were to make a map with the concentration of biodiversity across the globe, right here in Panama in the tropics would be one of the most biodiverse places.
Okay, first things first. What is biodiversity? I guess the first thing that I think of is that biodiversity is the total number of species in the world.
In the past, most estimates of species biodiversity were based on taking the number of known species from collections and then extrapolating based on the number of new species identified each year. The estimate was somewhere around 1.5 million. Then, in the late 70s, the work of Terry Irwin and Janice Scott, right here in Panama, opened the world's eyes to the concept that there could be a lot more species in the world. So, they fogged 19 individuals of one tree species with insecticide at different times of the year. Their results indicated that there were, say, 163 beetle species that existed on this tree and nowhere else. What if every tropical tree, all 50,000 of them, had similar endemic species? So from this data, they estimated that there may be close to 30 million arthropod species in the world. Then, people estimated that the total number of species may be as high as 100 million. So recent work has then re-examined this question. The newest studies think that maybe 10 million species is a safer bet. But in reality, no one knows. They're all just educated guesses.
But remember, biodiversity is more than just the total number of species. Population biologists are concerned about the genetic diversity as well. We see that, of course, every day in people.
The last form of diversity can be seen within ecosystems, like here in the tropical forests to the deserts, alpine tundra, and more.
But it's really scary just how fast we're losing biodiversity.
Introduced species from around the world are decimating local plants and animals. Habitat loss is decreasing the amount of available land. Disease, like avian malaria in Hawaii, is killing local birds, and the chytrid fungus in the neotropics is devastating amphibian populations. But the question still remains, why is this important?
So there's many different reasons why we should work to preserve biodiversity, of course. One could be purely aesthetic reasons. Artists, for instance, have gone out in nature for ages to search for inspiration.
A lot of our medicines come straight from nature.
Of course, a lot of our food comes from nature too.
Finally, there's a lot of interactions between species and the environment that we just don't know about.
So a quick recap, biodiversity means more than species diversity, it means genetic diversity and ecosystem diversity. But we are losing biodiversity, and the question still arises, what can we do about it?
So you may already know, there really is no one way to protect biodiversity. But here in Panama, they have this great approach where they set aside tracts of land as preserve zones. And then, to protect that land from poachers, they have a team of guardabosques that patrol the reserve. The guardabosques are kind of like nature security guards.
Okay, so I'm going to suggest something else that you can do. Anybody that owns land can plant native species in their yard, because by putting native plants, you encourage other native species to come in.
You can also visit places that set aside land for tourism. Many communities have found great value in protecting tracts of land to bring in ecotourists. Every time you visit these places, you're helping to protect these areas. But hey, remember to do it eco-friendly.
Our Earth contains so much diversity, not just the number of species, but within genes and among ecosystems as well. And just remember, even with our growing population, each of us can make decisions every day to help preserve what's here.
So the first thing you need to do, of course, is explore, because if you don't know what's around you, you can't do anything to protect it. But that's where we can help. Each week we're going to be looking at a different plant or animal and following researchers that are answering important questions in science. So, until next time, I'm Rob with the Wild Classroom, and we encourage you to never stop exploring.
So we can classify biodiversity into 3 different types of biodiversity. Genetic diversity, which means genetic variation within a species. This measure of diversity looks at differences among individuals within a population or at differences across different populations of the same species. While as the species diversity, this looks at the differences between species. It means the best fits literal translation of the term biodiversity, number of different species in a particular ecosystem or on Earth. This type of diversity simply looks at an area and report what can be found there. So all kinds of species that can be found in a certain habitat. Third type of diversity, we call it ecosystem diversity. And the ecosystem diversity relates to the interaction between the different species that live in an area with their environment. So this term does not include only life, but also land, sea, air that support life. In ecosystem diversity, biologists look at many types of functional units formed by living communities interacting with one another. Species richness, as we just said, is the measurement of the number of species present in a place. The more species present in a place, the rich the place is. It takes into account the number of individuals of each species present, and it gives equal weight to those species which have very few individuals and those which have many individuals. Let's take an example. You have here the number of individuals of 4 different species in sample A and sample B. Consider sample A and sample B are 2 different habitats. If looking at the number of species, both of them have the same number of species, but so by comparison between these 2 samples, we can say they have the same species richness, because each sample or both samples have the same number of species, regardless the number of individuals of each species. Because looking carefully here, you can see that rose angel has very high individuals in sample A and very few ones in sample B. Species richness does not take this individual number of each species into account. On the other hand, species evenness, it's a measurement of the relative abundance of the different species making up the richness of an area. So it takes into account the relative abundance of individuals in each species. So looking at the same example, I can say both samples have the same richness as we have just said, but the total number of individuals is also the same in sample A and sample B, but the evenness is different, because looking carefully, you can see that catfish and zebrafish, they are much more abundant in sample A than sample B. On the other hand, the freshwater snail is much more abundant in sample B than sample A. But we can say that sample A has more evenness comparing to sample B, because the number of individuals in each species in sample A, they are all nearly or almost equal. Or we can say that sample A is more condensed, sample A is more diverse. So we can consider that a community dominated by 1 or 2 species is considered to be less diverse than the one in which several different species have a similar abundance. The biodiversity index, it helps to quantify the biodiversity of a habitat. Biodiversity of a place is expressed as a numerical value, and it takes into account both species richness and species evenness. In order to calculate the biodiversity index, we need to have 2 values first. First, capital N, which refer to the total population size, and the small n, which refer to the population size of each species. Let's take an example here. We have 2 samples of 2 different communities in 2 different areas in the world. Community A and Community B. Community A has a relative abundance of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So the abundance here is 10. Species richness, so how many different species are existing here? 1, 2, 3, and 4. 4 species richness. While as in Community B, looking for the abundance, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10 organisms here. Species richness, the number of species, 1, 2, 3, and 4. So both communities have the same number of abundance and the same value of species richness. So why do they look different? They look different due to the difference in species evenness. The distribution of species, we can say it's more even in sample B than sample A. Or we can say the diversity in Community B is high, while the diversity in Community A is low. But how we are going to calculate diversity? We need a quantitative value for diversity. Here you need to apply the formula of diversity index. The formula is saying that D, which is D equal N into N minus 1 over sum of small n into n minus 1, while the capital N refers to the abundance. So I can say that n equals 10 in each of the two samples, while the small n is the total number of species, a total number of individuals in each species. Okay, so the small n here in sample A for the first bird it's 1, 2, 3, 4, 5, 6, 7, and the second one is 1, and the third species is 1, and the fourth species is 1. To calculate the diversity index, you need to develop this kind of tables, which shows all the values given in a more ordered way. The first one, you write the species, okay, the blackbird, kiwi, secretary bird, and the chickadee. In the abundance, you're going to write the abundance, the number of organisms in each species, and then find the value n minus 1 and n into n minus 1. Okay, and the last one is showing the total. The total abundance is 10. N minus 1, capital N minus 1 is 9. N into N minus 1, 10 into 9 equals 90. Okay, and then work out the formula. Okay, 90 over 42, because here it will be like 42 plus 0, 0 plus 0. So it's 42, equals 2.14. Now we need to analyze what does this value tell you about the degree of biodiversity or the level of diversity. So low species diversity means there is fewer successful species in this habitat. The environment is quite stressful to those organisms. The food webs are quite simple. Any change in the environment may have serious effect on the ecosystem. Additionally, we expect that those living organisms live in extreme environmental conditions. The ecosystem is unstable. They are very susceptible to any change. They have unfilled niche, means that if any incoming species entered this ecosystem, it will be welcomed, and it's going to overpower the existing competing species, and of course it will have a drastic effect on the balanced ecosystem there. While if you have very high value of diversity, this expects that you have very high species diversity, and this means that you have great number of successful species, means that they have more stable ecosystem, more ecological niches are available, the environment is less likely to be hostile, contains complex food webs, environmental change is less likely to be damaging to the ecosystem as a whole. They have a high level of productivity. The rate of photosynthesis will be very high. So the rate of growth and reproduction of all living organisms there will be very high. Here, introduction of new species into that ecosystem will almost have no effect. Causes of biodiversity loss. Why the biodiversity might be lost in an area? Due to many events like natural events, flooding, volcanic eruptions, also human activity like burning forests or destruction of forests, which cause disasters. Smaller and isolated ecosystem is more vulnerable to disasters. If your ecosystem is small, has few species occupied by living organisms, we said it's more vulnerable to be damaged due to human activity or due to natural events. But when to measure the biodiversity? To measure the biodiversity, you need to look for the time of the day and the season you are counting the biodiversity. Time of the day is very important because you may find some organisms in the morning and you can't see them in the afternoon. Secondly, the season. Some organisms like birds or fish, they migrate from one country to another due to change in the temperature or in the daylight. So when you are counting the biodiversity, you need to look for the time of the day, which time is more suitable. You can find the permanent organism that live there. And also you need to look for which season, which is occupied by the most varieties of living organisms. Biodiversity within a species or genetic diversity. We said that the biodiversity can be described on many different levels, on species diversity, genetic diversity, and ecosystem diversity. We have just described the species diversity, and we said that we use the diversity index to quantify this value. So here we are going to look for another term for diversity, which is genetic diversity. Genetic diversity is the diversity between the population of the same species or between individuals in a population. Here, the main kind of diversity here is caused by variation. And the main cause of variation is mutation. And when you are looking for a population of living organisms, we are not looking for what each individual organism have of genes, but we are looking for the gene pool. What is the gene pool? Gene pool is total of all genes in the genome. It involves all variants or alleles of a gene. It can be distinguished by using the DNA analysis, which is going to be used to measure the genetic diversity. What are the causes of genetic diversity? We said the main cause of genetic diversity is mutation, which result in a change in allele frequency. Some mutation result in adventitious features. So if the mutation result in adventitious features, is going to be selected by the ecosystem and is going to multiply and reproduce, then the allele frequency would increase in the population due to natural selection. This eventually will result in emerging of a new species. While if the mutation was disadvantageous, so natural selection is going to remove this allele from the gene pool. The process of natural selection is going to be repeated and repeated in every generation. So this allele will remain at very low frequency and might be completely removed from the population. This also may lead to emerging of a new species which lack the phenotype of that particular allele. But how to measure genetic diversity within a population? We said that we are going to analyze the DNA. Analyzing the DNA will give you a pattern of some similarities and differences of particular regions on the chromosome that we call them, if you remember, VNTR, variable number tandem repeats. Those patterns are going to analyze the degree of heterozygosity in the population. Just to remind you, the homozygous and heterozygous, homozygous individuals, they have the same allele exist on the same locus on a pair of a chromosome or on a homologous pair. If we have homozygous individuals that have the same allele on the two chromosomes or on the homologous pair of the chromosome, is going to appear as just one band of DNA, while the heterozygous individual will show two bands. So by counting how many two bands you are given on the DNA analysis, you'll be able to calculate what we call heterozygosity index. Heterozygosity index of genetic diversity. We have an individual that lives in a population. If this is the proportion of the heterozygosity of all alleles in this individual, so I can say that we have only one heterozygous allele two heterozygous allele out of 4. So the heterozygosity index here equals 0.5. The percentage of loci at which an individual is heterozygous is 0.5. Looking at the population, here we have 1, 2, 3, 4, 5, 6 heterozygous individuals. So the average heterozygosity in this population, you will divide the number of heterozygous individuals over all, so you will get it 0.461. What high heterozygosity index means? High heterozygosity index indicates high level of genetic variation and indicates a healthy population, while low heterozygosity index means that the population is in trouble. To calculate the heterozygosity index, as we said, we divide the number of heterozygous individuals over the total number of individuals in the population. Let's take another example. You have a frog here, and these are the pattern of heterozygosity and homozygosity in the population of toad. You can see here, this is homozygous, heterozygous, because as we said, the DNA band in homozygous individual is only 1, while it's 2 in the heterozygous one. So we are going to count how many heterozygous individuals divided by the total number of individuals in the population. So I have here 1, 2, 3, 4, and 5 over the total number, which is 23. So 5 over 23 equal 0.217. This is the heterozygosity index. If the value is less than 0.5, so we consider this value is low or quite low. It suggests the low rate of heterozygosity, meaning that the toad species are unhealthy and might be at risk of getting extinction or being endangered. We have an example here of an endangered species of organisms with the cheetah genetic diversity. You can see here that this is over time, and here is the population growth in 3 different countries, in Europe, Africa, and in the whole world, in the period between 1750 to 2150. You can see that the rate of the population growth is very low, is kept very low in Europe. It starts to increase only in 1999 in Africa, but in the whole world, it is starting to increase from 1950 onwards. If we analyze the cheetah genetic diversity, you can say that 99% of the cheetahs have the same genes. They have the same genes, means very low genetic diversity. Why they have very low genetic diversity? This might be attributed to so many factors, like low survivorship and poor sperm quality, greater susceptibility to disease like viruses, because most of the cheetahs in the world, they are distributed only in very few areas in the world, so they are going to be affected by the inbreeding consequences. And the inbreeding results in more of the succession or declining of the gene quality, low survivorship, poor sperm quality, greater susceptibility to disease like viruses. But the real survival problems faced by cheetahs, they say, are more of human-created sort, and loss of habitat and loss of prey. So mean that cheetahs are suffering from 2 different ways, one due to natural inbreeding case, and secondly due to human activity. Both those factors are going to decline the cheetahs population worldwide. But commenting on the graph, as the population was very low until 1950 worldwide, but they start to increase onwards, this might be attributed to conservation programs that have been established since about 50 or 60 years. You can see here some models of molecular phylogeny that can help to be used to highlight areas that need protection, also provides a way of monitoring changes. Look at these colors. These colors represent the areas of very high and very low genetic diversity. So these maps show biodiversity of bees around the world measured by species richness and the genetic diversity. Islands of Hawaii. There are 8 isolated islands of Hawaii that have been separated long time ago, and they all share some features. First, they are a very isolated system, or we can call they have common endemism. They have great deal of biodiversity in terms of species richness and species abundance, but they have low genetic diversity. There are no mammals except bats. The region is considered as a biodiversity hotspot, means it has a wide variety of species. Then it was saying that there were 7 species of founder organisms that have been introduced into the region long time ago. They are adapted and evolved to take the advantage of different ecological niches that were available to them. As a result, 400 different species have been evolved from those founder species. By DNA analysis, scientists now are sure that they are very closely related. So even though we have so many different species and they vary morphologically, but by the DNA analysis, or we can say molecular phylogeny, has been proved that they are all related to each other in spite of the variations between them. Now we are done with the lesson and we are going to solve some questions. We are going to start with the checkpoint questions in the textbook. Question 1. The term biodiversity is often used in the media simply to indicate the number of species of living organisms. Explain why it gives a limited picture. So in order to answer this question, first you need to refer to the term biodiversity, that it has different points of view: species diversity and diversity within a species, which we call it genetic diversity, and ecosystem diversity. You're going to see the answers in the next slide. Then, question 2, in a small area of European woodland, a group of students counted a number of species present in a measured area, numbers of individuals of the different species. Here are the number of organisms collected from each species, and you are asked here to calculate the index of diversity for this area of the woodland and decide whether you think it has a high or low biodiversity. And then you are going to use information supplied here and other sources and discuss why is it important to measure both species richness and genetic diversity to give a full picture of biodiversity. Let's look for the answers in the coming slide. We have learned some vocabularies during this lesson. First, diversity index, which means a way of measuring the biodiversity in a habitat using the formula capital N minus 1 over the sum of small n minus 1. Remember that N, the capital N, refers to the total abundance or the total individual of all species in an area, while the small n is the number of individual species in the area. Allele frequency, frequency with which a particular allele appears within a population. So the percentage of the allele within the total number of alleles in the population for a certain gene. Genetic diversity is a measure of the level of difference in the genetic makeup of a population. Heterozygosity index, a useful measure of genetic diversity within a population, which depends on the frequency of the heterozygous allele to the total number of alleles. So it equals the number of heterozygous over the number of individuals, the total number of individuals in the population. Measuring biodiversity, the answer of question 1 is because it doesn't take into account the genetic variation in a population. That's what we mean genetic diversity, and also the ecosystem diversity. So it's only to say that the biodiversity means the number of species in living organisms in an area. It's a very limited term. Question 2, you are given table for some species and the number of organisms collected from each species, and you are asked to calculate the diversity index. So first, the number of organisms, you know, it means the small n. Total number of organisms mean the capital N. Then you need to find n minus 1 and multiply it by n again to find n minus 1, n times n minus 1 for each species, and then find the sum of all, okay? And then use the formula in order to calculate the value of D. Okay, the diversity index here was 3.93. We consider this value is very high, meaning that the area is very rich in genetic diversity. Question 3. Question 3 was asking, using the information supplied and other sources, discuss why it's important to measure both species richness and genetic diversity to give a full picture of biodiversity. The number of species and the amount of genetic variation within species are both important. The number of different species and the number of individuals in each species is key information for understanding an ecosystem and how it can best be conserved. However, genetic diversity is also important. It gives an indication of how robust a species will be at surviving changes or not. Using both measures is the best way to understand the biodiversity. Now we are done with our lesson. Thank you very much.